those biceps. Okay.
Can I start, sir?
Yeah, shall I start? Right. Oh, okay, perfect. Hmm. Okay. Yes, good evening, everyone. This side Dr. Abhijeet, and today we are gonna discuss about this mock paper discussion. Yeah. So whatever topics we have completed in this week, like we will be discussing some important questions, like with respect to that. Because we have already discussed the topic in this entire week, I will be just focusing on how to, what points to be included in the answer and how to write the answer. That typical format is very, very important, right? So yeah, you already know this is the exam mastery live series that we have initiated from June 1st, right? We have initiated from June 1st, right? So yes, we are revising all the important concepts in a very crisp, conceptual and concise manner over here, right? So guys, let us just yeah. So before starting with that, let us, you already know about the core features of the Oracle app, like the live classes are there, right? After that, if you miss the classes, you can just go onto the app to view that. The recorded classes are there. The notes are curated in the form of Q&A, the same pattern like which like it is asked in the exam, right? Now the next thing, yeah. This is the basic subscription plan. If you want some additional discounts, guys, you can just refer my code that is Dr. Abhijeet 20 for some additional discounts. Now, guys, like I have included the questions over here, right? So the very first thing, like I think this topic has been discussed in this particular week. The first question that carries like 10 marks always like no start with certain long questions, guys. Start with long questions in the exam that would help you to uh like give you confidence also like that you have completed one of the longest question in the exam, and that is one, the very first question that is of 10 marks. Explain the structure, function and clinical significance of plasma proteins. Now guys, before starting that, you should know about the plasma proteins. We have already discussed this earlier in my lectures also. We have albumin, globulin, alpha 1 globulin, alpha 2 globulin, beta globulin, gamma globulin, fibrinogen. All these proteins are there, right? The plasma proteins, the major plasma proteins are alpha, albumin, globulin, and fibrinogen. Okay. Now, guys, like coming on to the structure, right? Coming on to the structure, guys, these are the basic plasma proteins that are being found, right? Apart from that, guys, you have to like know about the functions, guys. They have multiple functions in our body, right? So you just have to mention like the protein function, right? About later you have to like the most abundant plasma protein, that is albumin, and that is something very, very important, the osmotic pressure and the water balance that it helps to maintain, right? It plays a crucial role in maintaining the fluid balance in the body, right? So you already know the albumin, that is one of the plasma proteins. What is the clinical significance of this albumin, guys? It helps to regulate the amount of fluid in the blood vessels, preventing excessive fluid buildup in the tissues. It creates oncotic pressure. So basically, it helps to maintain the oncotic pressure, guys, right? So it prevents the fluid from accumulating in the tissues, which can lead to edema. So one of the clinical significances, these proteins, like one of the important proteins that is the albumin, it will never lead to this edema formation. So that is something like you have to write about this, right? Albumin is also help in maintaining the blood pressure, one of the most clinical significance, right? It exerts an osmotic pressure of about 25 mmHg. Okay, so it helps to maintain a proper water balance. Apart from that, right, we have other like functions and significances, like for example the immune function, right? So these plasma proteins basically they help have a very crucial role in the immune function, immune system, guys. So guys, you have to give like proper headings no before each like this part, and you have to just discuss it with respect to that. So yeah, So yes, giving proper headings and like giving answers and points will be like something which you have to like write in this format only. No one is going to like watch your stories written. So there will be to the point answers, and like it should be in the format like that. It should be like discussed in points, guys, right? So we have like three types of globulins: alpha, beta, and gamma. So basically, they transport and bind various substances in the blood. We've all discussed this, right? So that is about the immune function. Apart from that, we have buffering action also. That's one of the most clinical significance of these plasma proteins. So there are multiples, and some of them you have to write, like especially about in this question, the albumin and the globulin. That is something very, very important. And you have to write about the osmotic pressure. If there is clinical significance, please mention about the osmotic oncotic pressure maintenance by the albumin, and the globulin immune function is very, very, very important, right? So yes, that is something you have to mention. Now, even the blood coagulation, even you know that fibrinogen is basically a plasma protein which has a crucial role in blood clotting. That is again one of the most important clinical significance, right? So that is in this way, like you have to answer the question, right? You have to write certain headings, like what are the clinical significances, which plasma protein is like responsible for which important function in our body, and that is you have to explain a little bit about that because it's a 10-mark question, right? So that is about the first question, guys. Now, actually, all the questions are, guys, just mixed up, so we are just gonna discuss it like randomly. I've picked up questions from here, right? So yes, another question says task describes the genetic basis of thalassemia, guys. Now, guys, you already know that thalassemias are the abnormal hemoglobin caused due to altered subunit makeup, right? So basically, there is absence of one or more alpha or beta chains of hemoglobin. So we have already discussed this, right? There are four copies of a globin gene, two on each chromosome 16, and two copies of globin gene, one on each chromosome 11. Now, obviously, if there is any mutation, guys, if there is any mutation in these particular like point mutations are occurring, that can lead to thalassemia. So basically, they are classified into alpha thalassemia and we have beta thalassemia, and you have to like tell the subtypes and you have to tell how they are caused. You have to tell the genetic basis. So alpha thalassemia, it is caused due to defect in production of the alpha chain. So it can be alpha thalassemia silent, trait, minor, and major, right? So basically, if all the four genes are absent, globin genes are absent, obviously it is alpha thalassemia major. Absence of three alpha globin genes, alpha thalassemia minor. Absence of two alpha globin genes, alpha thalassemia trait. And absence of one of the alpha globin genes. So there are no clinical effects, just act as a carrier. Again, coming on to beta thalassemia, guys, it is caused due to defect in the production of the beta chain, right? So we have beta thalassemia major and minor. So if there are both beta globin genes are absent, right? So that is a homozygous condition, and that can be like classified as beta thalassemia major. Whereas if there is absence of only one beta globin gene, then it can be beta thalassemia minor, and it is an heterozygous condition. So you have to tell about like, yes, what is like which chromosomes are involved over here, right? Which chain, um, now how many genes are absent in which particular chain. So that is like, and this is all occurring due to like the point mutations, right? So that is something you have to remember. Now, again, the next question says, explain the function of the transferrin, guys. Explain the function of the transferrin. Now, you guys already know the plasma transferrin is a crucial, no, having a crucial role in this iron metabolism. Now, basically, what this transferrin do, we have already discussed this in our regular lectures, that it limits the level of free iron in the blood, right? It limits the levels of the free iron in the blood. Now, guys, you already know the free iron is dangerous. It carries the risk of triglycerides, free radical reactions, obviously, which sets off lipid oxidation and the destruction of thousands of molecules. Free radicals are basically, no, you already know these are like one unpaired electrons, guys, one unpaired electrons, and they will be driven to steal electrons from every cell tissue, including the heart, pancreas, brain. So that radical damage basically can contribute to heart diseases, the liver diseases, and neurological issues. Now, what this transferrin does, it binds to all the circulating plasma iron. Now, in plasma, whatever iron is being circulating, it will go and bind to the this iron. Now, what it will do chelation of these iron. Okay, it will do chelation of these iron. It will make it soluble, non-toxic. Okay, it will be delivered to tissues. Okay, so what will happen, guys? It will be suffering the functions of the rendering iron, okay, preventing iron triggered free radical damage. It will not allow this free radical damage to happen. Okay, it will help in the transportation of the iron. It will help in the transportation of the iron, guys, right? So that is like all these points that you have to include, guys. So how this transferrin is being um no, what is the clinical significance or the function of the transferrin in the body? You have to just mention about that. So transferrin can bind to two iron forms, right? So yeah, Fe3 plus binding sites, right? So everything you have to include in this particular answer. We have already discussed in the normal lectures. So that is regarding this transferrin. What are the functions of transferrin? Coming on to the next question, that is to describe the structure of a typical porphyrin ring. Typical porphyrin ring, guys, you already know porphyrin ring. Basically, it is made up of four pyrrole rings, right? It is made up of four pyrrole rings, basically joined by methylene bridges. So you have to write everything, guys. Methynyl bridges. So let me just correct it. It is joined by methynyl bridges, right? Now these methynyl bridges are nothing but this CH, double bond CH dash, right? So these methynyl bridges are there, right? And we have these side chains, guys. We have these side chains which you can see, right? We have these side chains which you can see. So basically, three types of porphyrins are there based on the side chains, right? So this is the normal structure of this porphyrin ring. Okay, normal structure of this porphyrin ring. So basically, remember this porphyrin ring, it is basically composed of a pyrrole ring. So we have a pyrrole ring, guys, over here, right? It's a five-membered ring. It is a five-membered ring, guys. It is a five-membered ring. So that is something you have to like mention over here. So we can have Europorphyrin, we can have okay, based on the side chains, guys, you already know we can have different porphyrins. We can have Europorphyrin, we can have yes, coproporphyrin, we can have protoporphyrin. So we can have like different based on the side chains. So that we have already like discussed before also. Now, coming on to the next question, guys, they are asking explain the role of hemoglobin and oxygen and carbon dioxide transport. This is something that, guys, you already know. And I would advise if you can just draw before this, okay? In this question also, guys, this structure you have to draw, guys. Wherever the word structure is there, you have to draw the diagram for that. Wherever the word structure is there, guys, you have to draw the diagram in that case, right? Even in this, the trans role of hemoglobin and oxygen and carbon dioxide transport, you have to first make the flowchart, guys. You have to first make the flowchart, and then you can like write the relevant points according to that. So you already know the affinity of oxygen to hemoglobin. It is basically affected by this. Okay, low pH, high BPG, and CO2 present in tissues favors the T state. We have already discussed this R state, T state, right? So binding of oxygen, it is also regulated by the partial pressure of oxygen, right? Every 100 ml of oxygenated blood carries 5 ml of oxygen to the tissues, right? So binding of the first oxygen molecule to the heme unit of one subunit of the deoxyhemoglobin T state, right, causes conformational changes leading to an increase in the affinity. The second molecule binds more rapidly. So that is like if the first molecule is binding, the other will be creating cooperativity reaction no, to the other sites as well. So that is the elastic binding that we have already discussed. So that is like the transport of oxygen. Even you have to tell about the transport of carbon dioxide, guys. You already know 100 ml of deoxygenated blood carries 4 ml of CO2 to the alveoli. So CO2 is transported bound to the hemoglobin as carbaminohemoglobin, right? So yes, this is the points. But before that, if you can just make a flow chart resulting how CO2 is getting transported with the help of hemoglobin to various parts of the body, that will be like first you have to draw the flow chart in that case, and then you have to write these particular theories. Okay, now these Bohr's effect in the hemoglobin, guys. I think this is something we have already discussed this, right? We have a left shift, we have a right shift that is called as the oxygen hemoglobin dissociation curve. So it is plotted against the oxygen saturation of hemoglobin and the oxygen partial pressure. So in these questions, guys, you have to first draw the graph. Graph is like some way very, very important, guys. Graph is like very, very important in these cases, right? You have to draw the graph and you just have to write about this, right? So it is a sigmoidal. What is the shape of the graph? How this shift is affected? Right shift, it is because of so decreased affinity for oxygen, increased PoCO2, increased H plus ions, increased 2,3 DPG, and increased temperature. So these all favors the right shift. Left shift, what all components are like favoring this left shift. So that is something you have to like mention in the graph itself, and then you have to explain. Now coming on to the next question, guys, they are saying they explain the different types of the porphyras and their biochemical basis. Now, guys, you already know we have different types. Like we have the hepatic porphyras, right? Acute and intermittent porphyra, porphyra cutanea tarda, hereditary coproporphyra, variegate porphyra. Then we have erythropoietic, that is congenital from by birth, that is congenital erythropoietic porphyra and protoporphyra. So guys, what is the enzyme defect in these porphyras? You just have to like, we have already discussed this in hemoglobin in detail, guys. In hepatic, in acute intermittent porphyra, it is uroporphyrinogen 1 synthase, right? Porphyra cutanea tarda, it is uroporphyrinogen decarboxylase. Hereditary porphyra, coproporphyrinogen oxidase. Variegate porphyra, protoporphyrinogen oxidase. So that is how you have to like mention the different types, what are the biochemical basis defects, and what are the characteristics that the patient will be suffering from. So just in the form of, not in the form of a flowchart, just try to write it like one by one, the enzyme defect, the characteristics, and where it is taking place. Now, guys, the next question says, in today's mock paper, we have this, yes, we have this the structure and function of the hemoglobin. So firstly, the structure of the hemoglobin, you already know it contains the globin part, that is the protein part, and a non-protein part, that is heme plus globin, right? So heme containing protoporphyrin 9 molecule with one iron atom, right, in the center. So iron plus protoporphyrin 9, it is like heme. So we have already discussed this. Then we have the globin part that is in the protein chain. Right, so that is you have to draw the structure of hemoglobin. Very, very, very important. You have to draw the structure of hemoglobin, then you have to explain these points with relevant to this particular structure. Okay. Now, apart from that, what are the functions performed by the hemoglobin? You already know we have exchange of gases, we have cooperativity, just I told you, right? All the functions that we have discussed in the classes, you have to mention over here, right? So that is here. First, you have to draw the diagram, then you have to write about that particular diagram. See, diagram should be technically correct, and then you have to write the functions of the hemoglobin, right? Coming on to next, that is the biochemical composition and functional role of lipids in the cellular membrane. So yes, you already know in the cellular membrane which lipid is responsible. That is the phospholipid making most of the part. And apart from that, we have cholesterol also. Cholesterol also, guys, right? So guys, firstly, you have to like describe the structure of this no phospholipid, also the composition, how this phospholipid is made. It is made up of phosphate plus lipid molecule, right? Phosphate plus lipid molecule, and you already know the it is basically a compound lipid or a complex lipid, and how this it is formed, right? So you already know it has a head, it has tails, right? So we have the hydrophilic and hydrophobic parts. You have to explain like everything. You have to draw the diagram, guys, right? Which is the hydrophobic part, which is the hydrophilic part, right? So yes, all the phosphate group, glycerol, it is like it is constituting the head part, the tail, right? The hydrophobic tails are there, okay, which are like so that is you have to remember. You have to first draw the structures, then you have to explain how these are arranged in the cell membranes, right? And how they are arranged as lipid bilayers. You have to like tell about each and every these components, and how they are like repelling the water which is inside the cell and which is outside. So that is something like if this is a membrane, guys, so they are arranged in lipid bilayer, right? So you have to like tell how these are like which one is inside, which one is outside. So whole structure should be clear, right? So that is something you can like maintain. And apart from that, cholesterol is something which helps in membrane fluidity. It helps in influences the membrane permeability also, guys, right? So cholesterol is also like you can mention over here. Next question is the structure and significance of triglycerides. Again, I don't have to tell you, it is formed of fatty acids plus glycerol, and glycerol is like the alcohol that we have drawn, and it's just removal of these molecules. And these are like we have the ester linkages, right? Ester linkages. So you have to draw the structure of these triglycerides. You have to draw the structure of these triglycerides. And then you will be like talking about the significances, right? So what are the significance of like triglycerides? So guys, the if you ask me, see, significance, like it can have multiple significance functions, but importantly, guys, you have to remember the levels of the triglyceride in the bloods are like, if it is under 150, it is normal. It is considered borderline. Above 200, it is high, and 500 is very high. Like high, see, triglycerides are used in the body because they provide energy. One of the important significance, right? Cells, they are naturally occurring component of the blood, right? They are can deposit in the as fat deposits. Now, if these are excess, what is the clinical significance, guys? If these levels are excess, no, they can lead to some syndrome called as metabolic syndrome. They can lead to the risk of diabetes, stroke, cardiovascular malformations. We have already discussed these high levels of triglycerides can affect the different systems in our body, right? Now, guys, you already know the glycolipids that are present in the cell membrane. The another question that we have to discuss over here, the glycolipids present in the cell membrane, guys. The glycolipids present in the cell membrane. And you guys already know we are talking about the gangliosides, guys. The glycolipids or the glycosphingolipids basically are the neutral compounds. They are composed of sialic acid residues. They are main components of animal cell membrane, and they are abundantly found in the neurons also. Right. So we have discussed these glycolipids in details, guys, right? Glycolipids in details. You have to tell about the structure, how they are like involved. Okay. So that is glycoproteins also, like we have already discussed in detail. So you have to tell about the composition, guys, the structure first, how this glycoprotein is formed. It is carbohydrate plus protein. Okay, so they are present in the lipid bilayer, how they are operating, no functions in the plasma cell membrane. So you should remember, yes, you have to tell about all the, yeah, it's like present on the lipid bilayer or the cell membranes. They can operate in the aqueous environment due to hydrophilic character, right? So that is something which helps in cell to cell recognition also. Yes. I think you will be able to answer all these questions because these are certain easy questions that we have already discussed earlier as well. Now, guys, let us just try to discuss questions from. Okay, let me open this. Yeah, let me open this. Yes. So we have discussed the first question, guys, right? The structure and the function of this. Yes, we have already discussed this, right? The first question explain the structure and the functions of these plasma proteins. Functions of carbohydrates in the body. I think I don't even need to tell you guys, right? So carbohydrates have like no, they have certain biochemical properties. And guys, we have already discussed these biochemical properties. You already know they are polyhydroxy, yes, alcohols or ketosis, right? We have already discussed. Apart from that, they have chiral or asymmetric carbons, right? Chiral or asymmetric carbons are there. Asymmetric carbons are there, right? They are generally manifested by the rotation of the plane polarized light, right? Plane polarized light. Now, they have the ability to form multiple hydrogen bonds. So these are the basic properties that we have all discussed in detail, guys. So that is something you have to mention all the properties over here. And guys, you already know the functions of the carbohydrates, right? The functions of the carbohydrates. So they help the save, yeah, the basic function help, basically they serve as primary energy resources, right? They provide energy, they help in the regulation of the blood glucose levels, right? They are helpful in performing various other functions also, right? Breakdown of protein molecules, even they help in eliminating ketosis. So that is like something you already know, right? Apart from that, guys, we have already discussed the structure of the hemoglobin, right? We have already discussed the structure of hemoglobin, done. Porphyra, done. Okay, lipids in the cellular membrane, you already know that, right? Okay, biological oxidation is something that we have already discussed in detail. You have to first draw the flow chart, guys. The flow chart is something very, very important over here. The flow chart that you have to draw, right? And then you have to tell how this is taking place in the inner mitochondrial membrane, right? Again, we have discussed the glycoproteins, Bohr's effect. I have already told you the graph is important. Then we have to tell the right shift, the left shift, the sigmoidal graph, and we have to tell like what are the factors which favor the right shift, which favor the left shift. We have already discussed this in detail, guys, right? Apart from that, structure and significance of triglycerides done. Role of hemoglobin in oxygen and carbon dioxide. I have already told you first try to draw a flow chart over here, right? And then try to explain this how the oxygen know it's coming from the blood to the peripheral tissues and everything, and how it is rotating. So just you have to like draw a schematic diagram, just a line diagram, that is like very important over here. Now, now the significance of the glycosaminoglycans, guys, we have already discussed this, guys. If you just remember, we have discussed the chondroitin sulfate, guys, the chondroitin sulfate. So this chondroitin sulfate, these glycosaminoglycans, no present in the chondroitin sulfate, okay, the heparin sulfate. So all these things you have to write like the significance of that. Now, the role of albumin in maintaining the plasma pressure, guys, we have just discussed in the big question, right? The long question. The same thing you have to write over here, how it is maintaining the, right? So importance of carbohydrates in cell recognition, yes. Now, guys, we have already like discussed all these questions, the typical porphyrin ring, the transferrin glycolipids. Okay, the high density lipids, guys, the high density lipids. And you already know if we talk about the high density lipids, it is basically, okay. Now, you all should know it is called as the good cholesterol, right? It is called as the good cholesterol, right? It is having the maximum protein content, least lipid content is there, least size is there, maximum apoprotein content is there, maximum apoprotein content is there, right? Even this is actually helping in the main function. It is actually the reverse cholesterol transport. So you have to tell about this reverse cholesterol transport that we have already discussed, guys, in detail. Reverse cholesterol transport, and this HCl, it is basically formed from intestine, like, and the liver, right? So these are the main points that you have to include while writing about this. So thalassemia, alpha beta thalassemia, we have already discussed, guys. So in case, guys, you like have to take the main points first, right? You have to write the headings, you have to include these points, and then you have to like write about this, right? So that is, yeah. Now, guys, these are something very, very like easy MCQs. Glucose to glucose 6 phosphate. You already know it is the hexokinase. Hexokinase. Now, hexokinase is something which requires Mg+2 enzyme, right? They will be like ATP will be broken down to form ATP. This hexokinase, it is basically like a housewife. I've told you between the glucokinase and hexokinase how to remember. The hexokinase is just like an housewife. Housewife is like working 24 hours, and glucokinase is like it's like mehman, it's like what we say, guest. So it will be working only at some places, but hexokinase is working like in all the cells of the body. So that is the main answer over here will be the hexokinase, which will help in the conversion from glucose to glucose 6 phosphate. Kinase, basically you know it is no, this group of enzymes basically helps to transport the phosphate group from one molecule to another, right? So yes. The primary storage of carbohydrates, that is something you already know, guys, right? Yes, the glycogen, right? The second one, hemoglobin is a type of, you already know it's a type of metalloprotein, right? Now coming on to the next one, the major component of cell membrane, you already know it's a phospholipids, guys, it's a phospholipids. The common precursor of steroid hormones, it is cholesterol, guys, right? The main function of albumin into the blood, it is maintaining the osmotic pressure. We have just discussed how it is maintaining the osmotic pressure, right? The rate limiting enzyme in cholesterol biosynthesis, we have already discussed, it is HMG-CoA reductase. Okay. The electron transport, again, it's the mitochondrial matrix, right? Now, mitochondrial matrix, which of the conditions is associated with defective hemoglobin synthesis? You already know it is the, yes. Hmm. Oh, sorry. This one. Primary site of ketogenesis, it is always the liver. Liver. Now that is something you have to remember, guys. Like these are all like very easy questions, but the thing is how you write it in the exam. It is like it matters a lot. So once you just go with the paper, it is like very easy. Just try to pick up the words like structure of plasma proteins. You have to tell about the structures, then functions, the clinical significance, write in the particular headings only so that it's like way easy. Okay, if you're talking about the clinical significance of albumin, so you have to write about oncotic pressure. If you're fibrinogen, how it helps in blood clotting, it's again a clinical significance of the plasma protein. So you have to just take one protein and their clinical significance, and then you have to explain that. So like two, three are like enough for that. So that is how like we can know get in like do this because we have already discussed all these topics. So I'm not like discussing it this. It just only matters how to write it out in an exam. Okay. So yes, that's all for like today, right? So we have already discussed it. So yeah, thank you.